causes of the cancer. Let us have a look over the chemical causes. See, among all the cancers across the globe which are prevalent in the human beings, 80% of the cancers are because of the chemical agents, or chemical causes happen to be the 80% causative agents for what? For the development or the cause of a cancer. So you will write down in your note that 80% of cancers, 80% of cancers among all the cancers are developed because of chemicals. 80% of cancers are because of chemical carcinogens. Because of chemical carcinogens. See the intensity of the chemicals which happen to be the causative agents for the cancers. Though cancer can be caused because of some physical agents or physical carcinogens, or carcinogens can be genetic, carcinogens can be environmental. But so far as all the causative agents in the etiology of the cancer are concerned, 80% of the cancers are being developed by the chemical carcinogens. So we will be having a thorough look over the chemical carcinogens. Now the chemical carcinogens can be of varied types. Some chemical carcinogens can be of the organic origin. Some chemical carcinogens can be of the organic origin. For example, dimethylbenzenthacene. For example, dimethylbenzenthacene. It is an example of organic chemical carcinogen. Okay, this is an example, and the chemical cause can be of inorganic origin as well. Inorganic carcinogens are also there. Dimethylbenzenthacene is an example of organic carcinogen. Okay, and there are also inorganic chemical carcinogens. And the examples of the inorganic carcinogens include arsenic. For example, arsenic. Okay. It also includes cadmium. Cadmium. What are these? These are the different examples of organic or inorganic chemical carcinogens. Okay. Now, how these inorganic or inorganic chemical carcinogens get their entry into the body of a person? Their entry can be varied also. The chemical carcinogens, you can ask me a question that how, sir, it is possible that dimethylbenzenthacene or arsenic or cadmium or any other organic or inorganic chemical carcinogen will get entry. How a person will allow such a type of the carcinogen to get its entry into the person's body? No one will allow, but still such chemical carcinogens get their entry into the body of the organism. Then there they develop the cancers, or then there they transform the cells. So the question is how they get entry into the body. They can get entry into the body through varied means. Here I will write down entry of such carcinogens. Entry of such carcinogens into body. Okay, into body can take place on occupational basis. Can take place, number one, on occupational basis. On occupational basis. For example, making use of asbestos, benzenes, etc. On occupational basis, entry can take place. Entry of different these carcinogens can take place. But the examples of such carcinogens which get entry into the body of a person on occupational basis, their examples include asbestos, benzenes, etc. Like asbestos, this chemical carcinogen is there. Like benzenes, these are a few examples. Similarly, the entry can take place via diet. If a person takes something which will be having such a chemical, the chemical would have been produced by any bacterium or by any biological agent, then we will take that eatable, and along with that eatable, such chemical which would have been the chemical carcinogen, it will get entry into our body through this food. So we can say the entry of the carcinogen can take place also via what? Via diet. Via diet. For example, you know about the aflatoxin B. For example, aflatoxin B. You might have heard about this aflatoxin B, which is produced by a fungus known as Aspergillus flavus. It is produced by fungus known as Aspergillus flavus. See, this type of fungus is located in most of the foodstuffs. When we talk about the peanuts, when we used to take peanuts, in most of the peanuts it has been seen that Aspergillus fungus is present on the peanuts or the seeds or the kernels, and it is this fungus, Aspergillus flavus, which has the ability to produce a metabolic product, and that metabolic product is what? Aflatoxin B. This aflatoxin B is a chemical carcinogen. So once a person will take the infected eatables like the infected peanut, and he will engulf the peanut, he will inculcate the peanut into her digestive system, and along with that peanut which would have been infected previously with what? With this Aspergillus flavus. When Aspergillus flavus would have been present over such an eatable, so for this aflatoxin B, it would have been very easy for it to get its direct or indirect entry into the body of the person, where it will act as a carcinogen and it will do its job. Okay, so main example of the food item over which Aspergillus flavus is present, main example is peanut. So we should take care of the peanuts or their clearness and or their quality while taking them or while using them. And next means of the carcinogens to get entry into the body is through drugs. You know, different people use different types of the drugs as they would have been recommended by their respective doctors. But some drugs have been seen and found methodologically and experimentally carcinogenic. And when a person takes such a drug which is carcinogenic, then he develops the chances, or any of the tissues in his body then develops the chances to get transformed and to get transformed into the cancer types of the cells. The examples of the drugs include diethyl cetibesterol. For example, diethyl cetibesterol. There are different types of such drugs which are carcinogenic, and diethyl cetibesterol is one among them. If a person takes along with his drugs such type of chemicals, then such a person becomes more vulnerable to develop this disease or this disorder. And sometimes the cause can be lifestyle basis. See, in someone's lifestyle has such a habit like making use of cigars or the cigarette smoking. If there is a smoker, then he makes use of the cigarettes. Then with that cigarette smoking, he is himself allowing the different types of the chemical carcinogens to get entry into his body and to develop the different types of the cancers. And in the lifestyle, we can put examples of cigarette smoking, and in the lifestyle, so far as Kashmiris are concerned, since during the winter times, we often make use of our fire pots or the kangarees continuously. We put such type of the kangarees or the fire pots have this close with our laps. And when we are continuously making use of such kangarees or the fire pots, we are exposing our body and our abdomen or our different organs like the gonads. We are continuously exposing them towards the heat radiations, and the continuous exposure of that heat can lead to the transformation of the cells, especially in the males or in the male gonads, to get transformed into the cancerous type of the tissues or the cells. So I will write down here use of kangarees. Use of kangarees in the lifestyle can also lead to entry of carcinogens into one's body. Though the heat radiations are the physical carcinogens, but this lifestyle can lead to easy entry of such carcinogens into our body. So such type of cancers which develop because of the continuous exposure to the heat radiations by one's body are mostly found in our Kashmir. Now, how we can say that a chemical, that a drug which is being used is carcinogenic? This is a question. There are different types of chemicals which are present in the pesticides, which are present in the fertilizers, which are present in the drugs. But now the question is how we can assess, how we know that among different types of the chemicals, there is a particular chemical or there are some particular chemicals which are what? Which are carcinogenic. For that, we have to conduct a test known as Ames assay. Known as Ames assay. So what is Ames assay? It is a laboratory test. It's a lab test used to conduct, used to know, which is used to know carcinogenicity, which is used to know the carcinogenicity of a chemical. See, if a person asks you that across the globe drugs are being used, across the globe fertilizers are being used, across the globe there are millions of types of the chemicals, but how we can know that a particular type of a chemical is carcinogenic? For that, we have to conduct this Ames assay laboratory test. In this test, we are making use of a particular bacterium known as Salmonella typhimurium. Salmonella typhimurium. I will let you know that how lab technicians are making use of the Ames assay to check the carcinogenicity of a chemical. See, this is a bacteria. Okay, it is wild strain or it is normal natural strain is not having the ability to synthesize histidine. Okay, to synthesize histidine. Listen, Salmonella typhimurium, which is roughly called as S. typhimurium. This very bacterial strain is not having the ability to synthesize what? To synthesize the histidine. So, if in normal in vitro circumstances, if you will go for the culturing of such bacterial strains, okay, you have to put the histidine as nutritional supplement, as nutritional supplement in the culture. Then when such bacterial strains will get the histidine with the nutritional supplement, since they are not having the ability to synthesize this histidine by themselves, they will get this with your nutritional supplement and they will make use of the histidine. These are normal circumstances. Usually, we mark these types of the Salmonella typhimurium as STHIS negative. It means Salmonella typhimurium histidine negative strains. So here, these are ST histidine negative strains. Now, what you have to do in the Amy's assay, you will take different chemicals. Say here, you will take one type of chemical named as 1, another type of chemical named as 2, another type of chemical named as 3. Okay. Now, with the nutritional bar, you will inculcate this type 1 of the chemical. Say this type 1 type of the chemical is one thing, and this type 2 type of the chemical is arsenic, or this type 3 type of the chemical is cadmium. This another type of the chemical which you have to inculcate into the nutritional bar is this dimethyl benzothioene. You have taken different types of the chemicals and you will use them one by one. First you will use say number 1, and you will put number 1 type of this chemical along with the nutritional supplement, and you will inculcate that into this. But when you will inculcate this one type of the chemical in with the nutritional bar, at that time you need not to inculcate the histidine. Okay, you have put this chemical in the nutritional bar to culture this Salmonella typhimurium. Since you know that Salmonella typhimurium is not having the ability to synthesize what? Histidine. When you have used this chemical but not histidine, and after culturing these bacterial strains for one generation or for the next generation, you will see these Salmonella typhimurium, if all the strains are ST histidine negative, histidine negative, it means that this very chemical is not what? This very chemical is not carcinogenic. Since this chemical has not lead to any type of mutation in the DNA or in the genetic material of this very bacterial strain, which will have lead to formation of this very product. Had there been mutation, had there been change in the DNA, had there been change in the genetic material of these bacterial strains with this chemical, then they would have achieved the ability to synthesize histidine. But since you have checked their different types of the bacterial strains and their different generations, all of the strains happen to be ST histidine negative. Now, you will conduct the next test and you will make use of type 2 chemical. Okay, when you are making use of type 2 chemical along with the nutritional supplement, except histidine for culturing of such type of Salmonella typhimuriums, and then you are after some time checking out one next or the next generation of what? Salmonella typhimurium, and you are seeing that there are some types of bacterial strains which happen to be ST histidine positive. What does that mean? That means this chemical was having the ability to transform the genetic material of the Salmonella typhimurium in such a way so that their changes, their mutations have lead them to achieve the ability for synthesizing what they could not have achieved previously. What is that? That is the ability to synthesize histidine. When they have attained the ability to synthesize histidine, then such type of strains are called as Salmonella typhimurium histidine positive. So, this chemical is now declared to be carcinogenic. Now you will declare this chemical to be what? To be carcinogenic. So similarly, with respect to other types of the chemicals, different such tests are being conducted to check the carcinogenicity of the respective chemicals. Once, by making use of any type of the chemical, any type of the drug, the Salmonella typhimurium achieves the ability to synthesize the histidine, then such chemicals are said to be what? They are said to be the carcinogenic. Okay? Almost 90% of the tests, 90% of tests to check carcinogenicity, to check or to test carcinogenicity of different chemicals are being conducted by this lab test, what is known as ME's assay. Okay? And after carcinogenicity of a particular chemical or a drug is tested by this test, and then the carcinogenicity can be tested for its final trial on animals. Like mice are there, monkeys are there, or other larger animals are there. After testing the carcinogenicity of a chemical, then the final test is being conducted over the animals, that whether they can transform or they can lead to the changes in the DNA in among that animals. Now, these chemicals or these carcinogenic agents, now they can be further divided into two types. Number one, either the chemical carcinogens, either they can only promote the development of the carcinogenicity among the cells, or they can directly lead to act as initiating agents for the development of the carcinogenicity among the cells or the tissues. So, different types of the chemical carcinogens or other types of the carcinogens can be divided into two types, either initiating agents or promoting agents. Chemical carcinogens can be divided into promoting agents. They can be divided into promoting agents, or they can also be divided into what? Initiating agents. Initiating agents. I will repeat it. What is the chemical carcinogenicity of a chemical, of a drug, or of any other agent or a chemical is tested? Once its test comes positive, then such types of the chemical carcinogens or other types of the carcinogens can further be divided into two types. Number one is promoting agents. Number second is initiating agents. It means these carcinogens can act as initiating agents. It means they can themselves initiate the carcinogenicity within the cells, within the tissues. And some types of the carcinogens can be only promoting agents. They can lead to promotions of the cells which have already been transformed by these initiating agents. And an important example of the initiating agent is benzopyrenes. For example, you write down here benzopyrenes. Okay, benzopyrenes are said to be the initiating agents for the development of cancers or the skin of the persons. And croton oil for it acts as the promoting agent. Croton oil. This acts as an initiating agent to develop cancer over the skin. a person is making use of a drug or a medicine which is containing this very type of the chemical, benzopyrene, then this acts as an initiating agent for the epidermal cells, stratum, spinosum, and the cells, different types of epithelial cells which are located over the skin of the respective person. And they are initializing the process of transformation of the skin cells. And the same person is making use of another chemical like croton oil. After he has used this very chemical, then this croton oil will promote the transformation of the skin cells. The process has been already initialized by what? By this initiating agent, benzopyrene. Now, the same person has used croton oil over his skin. Then this croton oil will do what? It will promote this transformation process. Henceforth, we can divide all such carcinogens into two types of agents. One is initiating agent, for example, benzopyrene. Another is promoting agent, for example, croton oil. Among the promoting agents, phenobarbital's, saccharines are the another examples. Phenobarbital, another example. Saccharines are another examples. So, if initiating process would have been initiated in a person's skin, in a person's another body organ, then if the same person is making use of such type of promoting agent, then definitely the tissue or the cells or the organ will get quickly transformed into the malignant types of the tumors. And varied examples of the promoting agents of different types of the chemical or other types of the carcinogens include phenobarbital, saccharines, etc. Now, these were generally associated with the chemical types of the carcinogens or chemical carcinogens. Next types of the carcinogens are physical. Among the physical carcinogens, there are UV radiations, there are X-rays, there are continuous or exposure to the heat radiations. All such physical agents have lot of energy. When such energetic rays penetrate into the cells of a person, then within the cells there is genetic material in the form of DNA. And that DNA may get changed into another types of the strains due to point mutations, due to deletion type of the mutations, due to translocation types of the mutations. When such changes will take place in the chromosomes and the DNA of the respective cells, then definitely they have the ability or they have the chances to get transformed into the cancer types of the cells or tissues. So, powerful ultraviolet radiations, powerful X-rays, and continuous exposure to the heat radiations can lead to transformation in the chromosomes or in the DNA or in the genetic material of the cells, which can transform them into the cancer types of the cells or tissues. And so far as this continuous exposure of the heat radiations are concerned, I have already told you that the Kashmiris people are continuously exposing their bodies to the fire pots or the concretes during winters. When we continuously hug the very hot concretes or the fire pots during winters for even months, for the days, for the hours, and that continuous exposure to the heat radiations can lead to the transformation of continuously dividing cells of the germinal epithelium of especially the male gonads. Since human males have extra abdominal gonads, and when we keep closely hugged such types of the fire pots, then our gonads or male gonads get continuously exposed to the continuous heat radiations that can lead to the development of the cancers in the male gonads. such types of the continuous heat exposure can also lead to the transformation of the cells over the skin, and skin cancers can also develop there. So among the different types of the populations which are present across Jammu and Kashmir, it is the Kashmiri people who are continuously making use of such types of the fire pots or the concretes who have more chances to develop such type of cancers. And number third type is genetic, or number third type can be genetic. See, every person's DNA has two types of genes. One type of gene has the ability to guide the cell to become a cancerous cell. Such type of genes are called as oncogenes or cancer causing genes. Listen, every person's cell has such type of chromosomes or such type of genetic material in which such type of genes are present, which are having the ability to guide the cell with the process of transcription, translation, to get transformed into cancerous type of the cell. Such type of genes are called as oncogenes or cancer causing genes. But you can ask me a question, sir, when every person is having such type of these oncogenes, then every person should develop what? Every person should develop the cancer. But it is not possible that if we will go for a check or the test among the people, then every person will be having the cancer. No. Because in every person there is another group of the genes which are known as cancer suppressor genes. So if in a person's DNA one gene is oncogene, if that gene would have been expressed, then that gene after transcription and translation would have resulted in the transformation of the cell. But in every person, the another gene, what is known as CSG or cancer suppressor gene, that before the expression of the oncogene gets expressed and leads to formation of some suppressor substances which suppress the expression of these oncogenes, and then check this type of gene to get expressed and to transform a cell into the cancerous type of the cell. If in a person there is a defect in the cancer suppressor gene or there is expression of the oncogene, then the same person can develop cancer. It is because of this fact that if a person is not smoking, if a person is not making use of such type of the chemicals, if a person is not having such type of bad habits with which he would have been exposing his body to get entry of different carcinogens into his body. But still, every person has chances to develop any type of the cancer in his body. Why? Because every person is having oncogenes within his her cells. But every person is not developing it. Why? Because every person at the same time is having cancer suppressor genes. If any type of the mutation will take place in the cancer suppressor gene or in the oncogene, then such type of mutations can lead to the expression of the oncogenes, and whenever there is expression of the oncogenes, then definitely there is development of any type of the cancer in different types of the tissues or the cells of that very person. And when such genes are the causative agents for the development of any type of the cancer, then the next type of the causative agent for the cancer come into play, what is known as genetic cause. And number 4 is another type of the biological cause. For example, viruses. Sometimes infectants like viruses can be the causative agents for the development of the cancers. It was the Rouse who did this handsome job in 1911. Rouse. Rouse did this nice job in 1911 to state that viruses can be the causative agents of different types of the cancers. But his work was ignored. Then after in 1980s he was awarded Nobel Prize after a very long time. Till then people were not believing his statements and his experiments. But nowadays we came to know that yes, it is the different types of the viruses which can lead to the transformation of the cells. Since we know that the HIV can lead to Kaposi's sarcoma, since we know that the herpes simplex virus or herpes zoster viruses, they also can lead to development of the cancers. And there are many other types of the DNA or the RNA or the retroviruses which are having the ability to transform the cells. So by now we will conclude it here today, and in the next lecture we will be discussing the molecular basis of the cancer. And here I will repeat quickly today's topic or your board, in which we discussed the etiology of the cancer. In the etiology of the cancer, we came to know that different agents can be the causative. These agents or vehicles for the development of the cancers. There can be chemical agents, physical agents, genetic causes or the environmental causes. And chemical causes constitute more than 80% of the cancers which get developed by them. And among the chemical carcinogens, we can divide them into the organic types or inorganic types. And the example of the organic chemical carcinogen, is dimethyl benzothionein, and the example of the inorganic, this chemical carcinogen is arsenic, cadmium. And then we also came to know the means of entry of carcinogens into one's body. Means of entry can be occupational, for example asbestos, benzene. It can be through diet, for example aflatoxin B, which is being produced by Aspergillus flavus, which is located on different types of the foodstuffs like peanuts. And the causative agent for the entry or the cause for the entry of different types of the carcinogens can be drugs, for example diethyl cetibesterol, or it can be because entry can take place because of the different lifestyles like overuse of congrees by the Kashmiri people, or cigarette smoking, or any other type of the smoking, or making use of different chemicals or the drugs by the different persons who have been habituated with the drugs. And how to check the carcinogenicity of a chemical? For that, where we have to conduct a test what is known as Ames assay. In Ames assay, we are making use of Salmonella typhimurium. Since we know that the Salmonella typhimurium is not having the ability to synthesize the histidine, so we have to put histidine into the nutritional bath, which has to be served for these strains to develop or to grow, to culture them. But if we have to test the carcinogenicity of different types of the chemicals, we will make use of one chemical with the nutritional bath to be served to these bacterial strains, but we won't serve the histidine. If after completion of a few these reproductions by the respective strains, we will check these strains. If all the strains are Salmonella typhimurium histidine negative, it means the DNA has not been transformed, otherwise that would have achieved the ability to synthesize histidine. But if we are making use of some other type of the chemical like benzopyrenes are there, phenobarbitals are there, arsenic is there, cadmium is there, if we are making use of such these carcinogenic agents, and we are putting such carcinogenic agents or such chemicals with the nutritional bath to get fed, to get the food supplement or the nutritional supplement to the bacterial strains, and we are not providing at that time also the histidine. And then after completion of few generations, we are checking such strains of the Salmonella typhimurium. Some strains come histidine positive. It means their DNA has changed, their DNA has been mutated, and that DNA now has attained the ability because of use of that chemical carcinogen to synthesize what? Histidine. When ST histidine positive strains come, then we say that such a chemical is carcinogenic. Then the final carcinogenicity is being tested over the animals. And besides that, after checking the carcinogenicity of different types of the chemicals, we have divided such carcinogens into two types of the agents. Either they can act as initiating agents for the development of cancer, or for direct transformation of a cell or a tissue into the cancerous type, or we can divide them into the promoting agents. An important example of initiating agent of the this carcinogen is benzopyrene, and some examples of promoting agents are croton oil, phenobarbital, saccharinase. If over the skin a person is making use of benzopyrene, this benzopyrene will act as a shading agent for the person is seeking to develop or to get transformed into the cancer. And if the same person is making use of phenobarbital, saccharinase, or the croton oil over the skin, then these chemicals can act as promoting agents. Then the respective person's skin cells will get definitely transformed into the cancerous type of the cells. Then we looked over the physical agents, and among the physical agents we saw ultraviolet radiations or X-rays or continuous exposure to the heat radiations can also act as the carcinogens. And we also discussed some genetic factors which are acting as the backbones behind different types of the cancers. We saw that the human cells have different types of the genes, but there are two types of the genes which are having the ability for the development of the cancers. And one type of genes are known as oncogenes. Oncogenes, if they get expressed within a person's cell, then definitely that cell will be transformed into cancerous type of the cell. But that type of the oncogene is continuously being masked or that is continuously being checked by the expression of another type of the gene which is known as cancer suppressor genes. So if any type of the mutation in the cancer suppressor gene or in the oncogene takes place, then the person's cell can lead to the development of the cancer. And we also discussed some viral causes or some biological causes of the cancers in the etiology of the cancers. When we discussed some viruses, it was Rouse who in 1911 told us about the carcinogenic impacts of the different viral agents or the viruses. Then in 1980s he was awarded Nobel Prize when every person came to know that definitely different types of the viruses are having the ability to lead to transformation of the cells or the tissues, because this time we know that